I'm Dr. Denise Hurd, Director of Research for the U.S. Poultry and Egg Association. Today, I am delighted to be speaking with Dr. Ravi Kulkarni about one of his current research projects. Dr. Kulkarni is an assistant professor in the Department of Population Health and Pathobiology at North Carolina State University. Welcome, Dr. Kulkarni, and thank you for sharing your research with us today.
Thank you, Denise, for having me, and it's always a pleasure talking to you.
Thank you. So to get started, I would just like to provide the audience a bit of information about the U.S. Poultry Board Research Initiative Program, also called the BRI program. The BRI operates separately and does not compete for funding or resources with the Comprehensive Research Program. We release special requests for proposals on specific topics. These topics are chosen by the boards of directors of the U.S. Poultry and U.S. Poultry Foundation. The requests for proposals released by the BRI are very focused and specify the research questions and areas of focus sought in each research proposal. 2 board research initiative projects are funded each year up to $125,000 each. Ravi, your study is titled Development of a Probiotic-Based Recombinant Oral Vector Vaccine Against Clostridial Dermatitis in Turkeys. Please provide us with an overview of your project.
So, Dennis, first and foremost, I would like to thank the U.S. Poultry and Egg Association and its BRI program and its board members for funding this project, which is very helpful to carry out this project. And about this project, as we all know, the Clostridial dermatitis, also known as gangrenous dermatitis or cellulitis, is an emerging disease of poultry, particularly affecting the turkey industry with huge economic losses. And in fact, it is considered, it is ranked #1 turkey disease, impacting economically very huge to this industry. And the Clostridial dermatitis, also abbreviated as CD, sometimes I would use during this interaction, it is caused by Clostridial bacteria that are anaerobic, spore-forming, and toxin-producing. And 2 pathogens have been implicated in this particular disease. One is Clostridium septicum, the other is Clostridium perfringens. Clostridium septicum has been implicated as the major pathogen because most often it is isolated from the clinical cases. And that's why our project is going to focus on this particular pathogen called the septicum. Now, the problem that we have is, considering the devastating nature of this disease that causes skin lesions, sudden mortality within a short window upon infection, we don't have a vaccine. In such cases, vaccines are very well preferred to control this disease, which we don't have one. That's why our work will focus on one of the major toxins produced by the Clostridium septicum called the alpha toxin. And we would like to clone the non-toxic parts of this alpha toxin into a probiotic bacterial isolate called the Lactococcus lactis and produce a recombinant Lactococcus-based vaccine that can protect against this Clostridial dermatitis in turkeys. So that's the kind of an overview of this particular project in our efforts in developing a viable vaccine against this important disease in turkeys.
Thank you, Ravi. That was a very, very good overview. Could you share some of your specific objectives with us?
Sure. While an overall objective is to develop a probiotic-based vaccine for this disease, our specific first objective is to clone the non-toxic parts of this alpha toxin from the gene into a Lactococcus lactis vector to express a recombinant Lactococcus lactis bacterial vector. And we will then confirm that the non-toxic part is indeed has lost its cytotoxicity, wherein we will do some in vitro cell screening to make sure that this part is not toxic. Now, the second objective, once we have the vaccine in our hand, we would like to immunize the turkeys orally and test the efficacy of this vaccine using a turkey model of Clostridial dermatitis disease. That is the experimental challenge model. And once we do that, this particular immunization giving turkeys orally, we will assess the protection against this particular disease, looking at various parameters, and also we will do the immunological investigations to find out what are the possible mechanisms of immunity against this disease. So these are the three specific objectives.
Thank you. And can you share a little bit about your experimental design and the methods for the construction and development of the probiotic-based recombinant oral vector vaccine with us?
Sure. The experimental design and the methods would involve the techniques related to molecular microbiology, molecular biology, again, that's the branch of the bacteriology, and immunology, and importantly, it would also involve animal modeling. Now, I'll try to explain the whole, the project experimental design and methods as briefly as possible. First, what we would like to do is to identify the non-toxic parts of the Clostridium septicum alpha toxin gene, looking at the structure-functional relationship, looking into the genetic sequence of this toxin, and also from the available literature. Once we know that the non-toxic parts of these alpha toxins, then we would like to clone these gene fragments into a plasmid vector, which is a circular DNA. And once we produce a recombinant plasmid that can harbor the non-toxic parts of the alpha toxin, then we will take this particular plasmid and insert that into the Lactococcus lactis bacterial vector. I have to mention here, we have chosen the Lactococcus platform because this particular bacteria or this particular isolate is, it allows us to clone with much more ease, meaning it is highly genetically manipulable. So once we can manipulate this particular, the probiotic isolate that can have the Clostridium septicum, gene fragments in it, then we have this particular vaccine ready. While this goes on in the laboratory, parallelly, we will also establish an animal model, a turkey model of Clostridial dermatitis. So we do have some previous experience in establishing this model. And once this model is ready, we are going to take the vaccine that we have developed in the laboratory and immunize turkeys orally, and then we will test its efficacy against the Clostridial disease, using the Clostridium septicum organisms as our, challenging or challenge pathogen. So the protection assessment finally would involve looking into the skin lesions, basically the dermatitis and then necrotic lesions, and also the mortality. And we will also check the performance parameters. And finally, we will do the pathology, the histopathology on these, skin lesions, and also some immunological investigation that would involve looking at the immune gene expression using the real-time PCR, antibody response by ELISA, as well as cellular immune responses using some neat techniques such as the flow cytometry. So I think, this is as briefly as I can give on the experimental design and methods.
That was very good, very good. What do you foresee as some of the impacts of this work for the poultry industry?
So then is the first and foremost impact of this work is that our work is expected to yield an oral probiotic-based vaccine to control dermatitis in turkeys. And this particular platform can also be extended for use in broilers and broiler breeders where it is still a problem, right? And the second advantage of this vaccine is, hoping all goes well as expected, while this vaccine would generate an antibody response that can prevent the dermatitis. This is the vector Lactococcus lactis by itself is a probiotic. So what I would expect is that it would also provide some gut health benefits to the turkeys. And all in all, I think, our findings should be able to provide a sustainable value-added means for our poultry producer industry to grow the birds without much relying on antibiotics to control these important Clostridial diseases.
Thank you. And last question. Is there anything else related to this project or follow-up projects that you would like to share with our audience today?
Sure. I would like to highlight here our team expertise in various areas that are molecular biology, immunology, pathology, vaccine development, as well as poultry medicine. Our collaborator, Dr. Anil Thachil, who is a clinical microbiologist at the NC State Veterinary Diagnostic Lab, he has previous experience working with this, turkey disease of Clostridial dermatitis, and his collaboration will be an asset. Similarly, we have Dr. Rocio Crespo, who is an expert poultry veterinarian, diagnostician, and also has research interests in Clostridial diseases. And we also have Dr. Francois Uzel from the University of California, Davis, who is an excellent pathologist and an expert in Clostridial diseases in animals. So all in all, I would say that we have put together a brilliant team to carry out this work. And I hope my laboratory, which is targeted to develop alternatives to antibiotics that would include whether probiotics or the vaccines to control Clostridial diseases in poultry, will come up very solid in the near future, and we would have much more projects to do in our laboratory with the help from organizations such as yours, US Poultry and Egg Association, and also other funding sources. So it's really wonderful to work with you guys.
Thank you. Yes, it sounds like you have an exceptional team put together to undertake this work, and we will be looking forward to the findings of it. Again, I want to congratulate you on your Board Research Initiative funded project. The BRI program is extremely competitive, so congratulations again on getting one of those projects funded. And also, as always, we appreciate you taking time out of your busy schedule to give us an update and an overview of what you have going on, which I know you have a lot at any given time. So again, we appreciate your time today, and we wish you the best, and we'll be looking for your findings in the near future.
Thank you very much.
Thank you.